So welcome to Biomandscape. Today we are going to know the tight junction, which are also known as the occluding junction, and it a role in the intestinal glucose absorption. So the occluding tight junction, in the diagram we can see here, they contain a separate apical and basolateral membrane of epithelial cell, and it ensure the different type of transporter are correctly localized. For example, sodium glucose symporters, they are present on the apical membrane, and glucose carrier, that is a glut, on the basolateral membrane. So without the tight junction, the transported protein couldn't mix, disrupting directional glucose movement. So it prevent the backflow of the glucose, that is, it act as a barrier. The tight junction seal the space between the adjacent cell and prevent the substance like glucose, water and sodium from leaking back into the gut lumen via the paracellular route. And why it is important? Because it helps in maintaining the unidirectional flow of glucose from the lumen to the cell and from the cell to the blood, and then finally it increases the absorption efficiency. So it is selectively permeable. While tight junction it works like it blocks the charged molecule which are present, and it allows the small ion or water to pass selectively in other tissues. In the intestine, tight junction are highly regulated depending on the nutritional needs. So it maintains the transport direction, which keeps the apical and basolateral function different. It prevents the glucose loss, it stops the leakage in the intercellular space, and it supports the efficient absorption, that is maintaining the higher intercellular glucose of transport. So tight junction, if it is damaged, so it causes infection, so the glucose absorption is impaired and the gut will be leaky, and the leaking may occur. So certain toxin like cholera toxin, it disrupts the tight junction and it serves the fluid loss. So the process occur in the intestinal epithelium. It consists of polarized epithelial cell. So in this diagram we can see cell 1, cell 2 and cell 3. They are involved in active transport at the apical surface, that is facing the gut lumen, then facilitated diffusion at the basolateral surface which is facing the blood, and the tight junction which prevent the backflow, and glucose concentration gradient, it is high inside the cell and lower in blood and lumen. So the apical membrane facing the lumen of the gut, it have sodium driving glucose symporter. This is secondary active transport mechanism where the sodium moves down there, the concentration gradient into the epithelial cell, then the glucose is co-transported against its gradient using the energy from the sodium movement, and this Symport protein is shown on the apical surface, that is the top of the cell. Then cytoplasm of the epithelial cell, glucose accumulate inside the cell. It create a high intracellular glucose concentration. Then there is a basolateral membrane which is facing the blood. It is passive glucose carrying protein which facilitate the diffusion, and the glucose is transported down in the concentration gradient in the blood and no energy is needed. So this is passive transport. This protein is located on the basolateral surface. The tight junction, they are located near the apical surface between adjacent cell. It prevents the glucose from leaking back into the gut lumen and help in maintaining the cell polarity, that is different protein on apical and basolateral surface. So sodium potassium pump is implied here. Sodium that enter the cell is pumped out by sodium potassium ATPase on the basolateral surface, and it maintains the sodium gradient required for sodium glucose symporter to function. So glucose concentration gradient as shown on the right, the lower glucose in the gut lumen and higher glucose inside the epithelial cell we can see here, and lower glucose in the blood until it is absorbed. So it is a location from lumen to cell, glucose transport, sodium glucose importer is active, and from cell to blood it is facilitated diffusion, that is a passive transport. So the system efficiency absorb glucose even when its concentration in the gut is lower. Critical for it is important for maintaining the blood glucose level after a meal. So in this diagram we can see how the glucose is absorbed from the lumen of the gut into the blood through the intestinal epithelial cell, and it is transported, it is a transporter mechanism and the cell junction. So This process occurs across the intestinal epithelium. It consists of polarized epithelial cell, which is cell 1, cell 2 and cell 3. So which one is facing the gut lumen? The active transporter, at where it is in the apical surface. Then facilitate diffusion at the basolateral surface, which is facing the blood. Tight junction, preventing the backflow, and a glucose concentration gradient, which is higher inside the cell and lower in the blood and lumen. So how it is transported? In apical membrane, in cytoplasm of epithelial cell and basolateral membrane. So the apical membrane, it is facing the lumen of the gut. The cytoplasmic of epithelial cell, where the glucose is accumulated inside the cell, creating a high intercellular glucose concentration. What is happening here in the cytoplasm? There is a high intercellular glucose concentration. And in basolateral membrane, it is facing the blood. So it is passive glucose carrier protein. It is facilitated diffusion. So glucose is transferred down in the concentration gradient into the blood. So no energy is needed, and this is passive transport. So this protein is located on the basolateral surface. So tight junction, what is it? It is located where? Near the apical surface between the adjacent cell. What does it do? It prevents the glucose from leaking back into the gut lumen. It helps in maintaining the cell polarity. Different protein on apical and basolateral surface. So there is sodium that enter the cell in pumping out via the sodium potassium ATPase on the basolateral surface, and this maintain the sodium gradient required for the sodium glucose Symporter to function. So glucose concentration, how it is? Lower glucose is present in the gut lumen, higher glucose inside the epithelial cell, and lower glucose in the blood until it is absorbed. So lumen to cell and cell to blood. Lumen to cell, sodium glucose Symporter. Cell to blood, facilitated diffusion, which is passive. So the function, it is this system efficiency absorb the glucose when its concentration in the gut is lower. So critically, it is important for maintaining the blood glucose level after a meal. So what is the role of occluding junction, the tight junction? In the intestinal glucose absorption. So we have learned that it maintains the cell polarity. They separate the apical and basolateral membrane of epithelial cell, and this ensures the different type of transporter are correctly localized. For example, the sodium glucose Symporter on apical membrane and Glut on the basolateral membrane. So without tight junction, the transported protein couldn't mix and disrupt the directional glucose movement. It prevents the backflow of the glucose. That is, tight junction seal the space between the adjacent cell and preventing substance like glucose, sodium and water from leaking back into the gut lumen via the paracellular route. So this helps in maintaining the unidirectional flow of glucose from lumen to cell to blood and increasing absorption efficiency. It is selectively permeable. While tight junction block the large and charged molecule, they allow small ion or water to pass selectively in other tissues. So in the intestine, tight junction are highly regulated depending on the nutritional need, and its function is to maintain the transport direction, preventing glucose loss and support the efficient absorption. If the tight junction are damaged because of the inflammation or infection, the glucose absorption is impaired and leaky gut may occur. So certain toxin like cholera, it disrupt the tight junction and serve the fluid loss. So the occluding junction, they are also known as the tight junction. They are the specialized connection between the adjacent epithelial cell that form the seal to prevent the passage of molecule between the cell, that is a paracellular route, and they maintain the polarity of a cell by separating the apical and basolateral surface. They are found in epithelial and endothelial tissues and most prominently in the intestinal epithelium like kidney tubules, blood-brain barriers. So tight junction are formed by transmembrane protein that stitches the adjacent cell together. So there is claudins, the major sealing protein, occludins. They help in the barrier and signaling, and junction adhesion molecule, JAMs, and helps in stabilizing the structure. Zonal occludins, that is the ZO protein, which link the tight junction to the actin cytoskeleton inside the cell. So protein interaction in the extracellular spaces to create a continuous belt-like seal around the cell. So the function of occluding junction is preventing free passage of substance like iron, water and pathogen between the cell, and it maintains the selectively permeability, that is, it is a barrier function. Then it maintains the cell polarity by preventing mixing of membrane protein between the apical and basolateral surface, that is French function. Then signal transduction, it involves intercellular signaling and gene expression regulation. It helps the cell withstand mechanism stress in tight epithelial layer. So there is Leaky Gut Syndrome. It disrupts the tight junction in a gut and allow the toxin to enter the bloodstream. If there is a leaky gut syndrome, so for preventing it, we need this tight junction. Then for cancer, altering tight junction can allow cancer cell to invade and pathogen. For example, some bacteria and virus example, the Helicobacter pylori or Clostridium, it target the tight junction to enter the tissue. So two epithelial cells side by side at the top where the membrane meets. The tight junction protein like Chlorella and Occluding forms a zipper-like seal and preventing the substance from slipping between them. So each tight junction seal stand is composed of a long row of transmembrane adhesion protein which are embedded in each of the two interacting plasma membrane. The extracellular domain of this protein join directly to one another by occlude the intracellular spaces. So the major transmembrane protein in tight junction are chlorines, which are essential for tight junction formation and function and differ in different tight junction. A specific chlorine is found in a kidney epithelial cell. For example, it is required for magnesium to be reabsorbed from the urine in the blood. A mutation in a gene encoding this chlorine which result in excessive loss of magnesium in the urine. A second major transmembrane protein is tight junction is Occluding, so the function of which is uncertain. The chlorine and Occluding associated with intracellular periphery membrane protein, they are known as a ZO protein. So the tight junction is also known as zonula occluding, which anchor the strand to the actin cytoskeleton. So in addition to chlorine and occluding, ZO proteins serve other protein that can be found associated with the tight junction. So these include some of the regular epithelial cell polarity and other that help guide the delivery of component to the appropriate domain of a plasma membrane. So thus the tight junction, it serves as a regulatory center for multiple processing of the cell. So in vertebrates, septated junctions are the main occluding junction, more regular in structure than in tight junction. They form a continuous band around each epithelial cell, but their morphology is different because they interact with the plasma membrane and they are joined by the protein that are arranged in parallel fashion and in a regular periodicity. So protein called disc large, which is required, it form the septa junction in, for example, in Drosophila, and it is related to ZO protein. It is found in vertebrate tight junction. Then if there is a mutation flies that are defect in this protein, they does not lack the septate junction, but they develop epithelial tumor. This observation is suggested that the normal regulation of the cell proliferation in epithelial tissue, it may depend on the intracellular signaling that emanate from occluding junction. So a septate junction, a conventional electron microscope of a septate junctions between two epithelial cell in mollusk and the interaction plasma membrane seen in the cross section are connected by parallel row of junction protein, and the rows which have a regular periodicity are seen as a dense bar or septa, according to this particular diagram of cell communication book.